Assalamu alaikum. Today's topic is the introduction to the specific immunity. There are two types of immunity directed against specific organisms. First is a natural immunity, which exists in the absence of exposure to the organism, and second is the acquired immunity, which results either from the exposure to the organism, which is called as active immunity, or from preformed antibody made in another host, which is known as passive immunity. First, we will talk about the natural immunity. Certain species have natural immunity against a particular organism. For example, Shigella, the bacterium that causes dysentery, is unable to infect non-primates. Only humans and chimpanzees are affected. In addition, some racial groups are more resistant to certain organisms. Light-skinned people are one-tenth as likely to develop the disseminated form of fungal disease coccidioidomycosis as are dark-skinned people. The age of an individual is also a significant factor. Generally, very young and very old people are most susceptible to infection, but for some viral diseases, such as poliomyelitis, young children get inapparent infections, whereas old children and young adults contract severe diseases. The basis for the differences seen with polio is unknown. Now let's talk about acquired immunity. There are two types of acquired immunity: passive acquired immunity and active acquired immunity. Passive acquired immunity. Passive acquired immunity is temporary protection against an organism and is acquired by receiving serum containing preformed antibodies from another person or animal. Passive immunization occurs normally in the form of immunoglobulins passed through the placenta, IgG, or breast milk, IgA, from mother to child. This protection is very important during the early days of life, when the child has a reduced capacity to mount an active response. Passive immunity has the important advantage that its protective abilities are present immediately, whereas active immunity has a delay of a few days to a few weeks, depending upon whether it is a primary or secondary response. However, passive immunity has the important disadvantage that the antibody concentration decreases fairly rapidly as the proteins are degraded, so that protection usually lasts for only a month or two. The administration of preformed antibodies can be life-saving in certain diseases such as botulism and tetanus that are caused by powerful exotoxins. Serum globulins given intravenously have been used to treat impending endotoxin shock in patients with gram-negative sepsis, and as a prophylactic measure in patients with hypogammaglobulinemia or bone marrow transplants. In addition, they can mitigate the symptoms of certain diseases such as hepatitis caused by hepatitis A virus, but they appear to have little effect on bacterial diseases with an invasive form of pathogenesis. Active acquired immunity. Active acquired immunity is protection based on exposure to the organism in the form of overt disease, subclinical infection such as an infection without symptoms, or a vaccine. This protection has a slower onset but longer duration than passive immunity. An important advantage of active immunity is that a secondary response occurs. There is a rapid response of large amounts of antibodies to an antigen that the immune system has previously encountered. Active immunity is mediated by both immunoglobulins and T cells. Immunoglobulins protect against organisms by a variety of mechanisms such as neutralization of toxins, lysis of bacteria in the presence of complement, opsonization of bacteria to facilitate phagocytosis, and interference with the adherence of bacteria and viruses to cell surfaces. If the level of IgG, immunoglobulin G, drops below 400 mg per deciliter, the risk of pyogenic infections caused by bacteria such as Staphylococci increases. T cells mediate a variety of reactions including cytotoxic, destruction of virus-infected cells and bacteria, activation of macrophages, and delayed hypersensitivity. T cells also help B cells to produce antibody against many but not all antigens.